In the early 1800s, families across parts of Europe ate preserved meats and expected a simple meal. Sometimes, hours or days later, people collapsed into a frightening sickness. It began like ordinary food trouble, nausea, stomach pain, vomiting. Then it turned, eyes blurred, speech thickened, swallowing failed. Strong adults became helpless, not from fever, but from a quiet loss of control. The pattern repeated often enough that communities gave it a name: sausage poisoning. The label sounded almost casual, but the outcome was not. Some victims recovered slowly. Others died, fully aware, unable to breathe. Doctors tried the tools they had, purges, bleeding, herbal remedies, and saw little change. Nothing spread from person to person. Nothing looked like plague. The threat seemed trapped inside the food, yet it stayed invisible. Without a clear cause, fear followed everyday habits. A perfectly normal batch of sausage could be safe, and the next one could cripple. People argued over what went wrong: spoiled meat, bad spices, smoke, storage, weather. Every explanation sounded possible, and none held up reliably. What made it terrifying was the precision of the harm. The body did not rot or burn, it simply shut down, one muscle at a time. In the 1820s, a German physician named Justinus Kerner took these scattered sausage poisoning stories and treated them like evidence. He gathered reports, compared symptoms, and looked for a pattern that kept repeating. People did not just get sick, they lost control. First, small functions failed: swallowing, speaking, focusing the eyes. Then the weakness spread. Kerner started to suspect the problem was not infection or bad digestion. Something else acted quietly and consistently. Kerner argued that an unseen biological poison did the damage, and he placed the target in the nervous system. That idea mattered because it explained why the mind stayed clear while the body shut down. He described the paralysis with unusual precision for the time: drooping eyelids, dry mouth, blurred vision, choking on food, a voice that faded, arms and legs that grew heavy. The order of symptoms suggested a process, not random collapse. A hidden agent blocked the signals that made muscles work. As Kerner followed the cases to their worst outcomes, one fact kept returning: many victims did not die from pain or fever. They died because the muscles that moved air simply stopped. Breathing failed. The body remained present, but the command system went silent. Kerner could not name the exact substance, and he did not isolate a microbe, but he reframed the mystery. This was a nerve poison, not a curse of sausages. His careful symptom map turned rumor into a clinical profile others could test. In 1895, another food poisoning disaster struck, and this time the clues did not stay trapped in guesswork. People became violently ill after a shared meal, then slipped into a frightening weakness that spread through the body. The pattern looked familiar, but the cause still needed proof. Belgian bacteriologist Emil van Ermengem took the case seriously. He treated it like a hunt for a single hidden culprit. Van Ermengem traced the outbreak back to the contaminated food and started testing what remained. He did not just look for any germ, he looked for one that survived where air did not help it grow. Step by step, he found an organism that thrived without oxygen, an anaerobic bacterium. It matched the illness in a way earlier theories could not. For the first time, botulism connected to a specific microbe. That link changed everything. Botulism no longer floated as a mysterious curse of certain foods. It had a biological source that could be studied, tracked, and named. The bacterium later became known as Clostridium botulinum. It explained why outbreaks appeared in sealed or poorly preserved foods, where oxygen stayed out and danger stayed in. The killer was not the sausage itself. It was what grew inside it, unseen. In the early 1900s, the focus shifted from the bacterium to what the bacterium made. Researchers worked to pull the active poison out of contaminated material and separate it from everything else. That step mattered because it turned botulism into something that could be handled, tested, and compared. The danger no longer seemed vague. It became a substance, isolated, concentrated, and ready to measure. Once the toxin was concentrated, its power became impossible to ignore. Small amounts produced the same pattern again and again. Muscles weakened, swallowing failed, breathing stopped. The key discovery was scale. It did not take a spoonful. It did not take a drop. The toxin worked in amounts so tiny they challenged the instincts of experienced scientists. Minuscule doses killed. That realization changed the entire problem. Botulism stopped being only an odd kind of food poisoning and became a measurable threat with clear rules: potency, dose, and exposure. With a purified toxin in hand, researchers could now test limits, compare batches, and begin thinking in numbers instead of guesses. The poison had a new identity, defined not by mystery, but by precision. By the 1920s, botulism no longer looked like bad luck. Health officials treated it like a solvable problem. Home canning had spread fast, and so had the danger. Sealed jars created the oxygen-free conditions the bacterium needed. One mistake could paralyze an entire family. So authorities pushed clear rules: acid levels, pressure cooking times, reliable temperatures. The threat stayed real, but it started to look preventable. Standards turned into systems. Inspectors watched commercial canneries. Extension agents taught safe methods to households. Labels and pamphlets reduced guesswork to steps people could follow. And when cases still happened, investigators traced them with new rigor, linking illness to a specific batch, a specific process, a specific failure. That changed behavior. Canning stopped being a private gamble and became something public health tried to control. At the same time, medicine chased a second line of defense: antitoxin. Doctors could not reverse paralysis once nerves stopped working, but antitoxin could bind toxin that still circulated in the body and keep it from doing more damage. That meant speed mattered. Suspicion triggered action, not waiting. By the 1940s, botulism still terrified people, but it no longer seemed unstoppable. Prevention and rapid response finally narrowed its window. By the mid-20th century, botulinum toxin no longer sat only in medical journals and public health warnings. Military planners treated it as a new kind of tool. The logic felt brutally simple. If a speck could shut down breathing, then a controlled supply could threaten whole groups at once. That idea pulled the poison into classified programs, where the goal shifted from prevention to deployment. To make that possible, the toxin had to become predictable. Teams worked to produce it in quantity, purify it, and measure its strength the same way they measured explosives or fuel. They tested how it entered the body, how long it took to paralyze, and how little it took to kill. Safety procedures tightened, but the purpose stayed dark. Control over the poison became the point. Those programs exposed a chilling fact. A substance that once struck by accident could now be shaped into strategy. With the right handling, the toxin turned from a rare foodborne disaster into something that could be spread deliberately, silently, and efficiently. Even when international pressure pushed limits and secrecy grew, the message remained clear. Science did not just explain botulinum toxin, it scaled its harm. In the 1970s, a new kind of idea took hold in medicine. If botulinum toxin caused paralysis by blocking nerve signals, then perhaps that paralysis could be made small, local, and useful. An ophthalmologist named Alan Scott focused on a stubborn problem. Eyes that did not line up because certain tiny muscles pulled too hard. Surgery helped some patients, but it could not fine-tune muscle force. Scott tested a different approach. He used extremely small, carefully measured injections of the toxin directly into an overactive eye muscle. The goal was not to damage the muscle, but to quiet it. If the dose stayed controlled and the placement stayed precise, the muscle weakened just enough for the eyes to realign. A poison known for spreading through the body suddenly behaved like a pinpoint tool. Each trial carried an obvious risk: too much toxin or the wrong spot, and the effect could spread beyond what was intended. That danger forced a new discipline: tight dosing, careful observation, and repeatable technique. But the outcome was hard to ignore. Scott showed that the same biological mechanism that killed in food poisoning could also be directed at a single muscle and turned into therapy. The turning point came from control. In the 1980s, the shift from risky experiment to approved medicine depended on one thing: control. Regulators evaluated purified botulinum toxin products like any other drug, demanding consistent manufacturing, stable dosing, and clear evidence of benefit. Approval arrived first for narrow, specific disorders where the problem was obvious: muscles that pulled too hard, too often. The toxin no longer stood only for danger. It became a measured tool. Once a regulated product existed, clinicians began testing where that same precision might help next. They treated dystonias, in which muscle contractions twisted the body into painful postures, and they treated spasticity, where damaged nerve pathways left limbs stiff and resistant. The logic stayed simple: weaken the overactive muscles without weakening everything else. Outcomes mattered. So did side effects. Technique became part of the therapy. In the 1990s, use expanded again, this time into chronic pain conditions linked to constant muscle tension and involuntary contraction. The goal was not sedation and not numbing. It was interruption, breaking a cycle of tightness, strain, and misfiring signals. Clinics began to build dosing patterns, injection maps, and follow-up schedules because the effect wore off and had to be managed. A lethal poison had entered everyday medical practice, one controlled injection at a time. In the early 2000s, a shift happened fast. Botulinum toxin stopped sounding like a lab warning and started sounding like a brand. Under the name Botox, it moved into everyday language and ordinary clinics. People who never faced muscle disease now sought the same substance for smoother foreheads and softer frown lines. A drug built on paralysis became a cosmetic routine. The poison did not change. The context did. Demand surged because the promise felt simple: tiny injections, quick visits, visible results. But the simplicity hid the stakes. The dose had to stay precise, the placement had to stay exact, and the product had to stay properly handled. As more providers offered it, questions followed close behind. Who had enough training to inject it safely? How often was too often? And what happened when the toxin spread beyond the intended muscle? Reports of bad outcomes kept the debate alive: drooping eyelids, uneven expressions, trouble swallowing in rare cases when effects travelled farther than planned. Regulators and manufacturers tightened rules, warnings, and labelling, while clinicians argued that controlled use remained safe for most patients. At the same time, the black market version grew as a shortcut around cost and oversight. Botox became both normalized and contested. A notorious poison now lived in the appointment book. Today, botulinum toxin lived 2 lives at once. On paper, it remained one of the most lethal poisons ever measured, the kind that could not be treated casually because the margin for error stayed razor thin. But in clinics, it also served as a routine tool, ordered, stored, and prepared like any other prescription product. That contrast defined the modern era. The danger never disappeared. the handling simply changed. Control became the whole story. Purity, dose and technique decided whether the toxin harmed or helped, and modern use depended on systems that forced those decisions to stay consistent. Manufacturers produced standardized vials. Clinicians followed strict protocols. Regulators tracked indications, reporting and warnings. It meant the same biological effect that once spread unpredictably through food now got limited to a small, chosen set of nerves. Precision replaced surprise. That precision created a new kind of risk. Not mystery, but misuse, mistakes, off-label ambition, counterfeit supply, and bad training all carried real consequences because the substance itself stayed unforgiving. Yet the public image shifted anyway. People associated the name with smoother skin or eased muscle spasms, not sudden paralysis and death. The split reality held a deadly poison and a regulated staple, far from the helpless confusion of sausage poisoning.